I missed was at some point you guys read about DNA mutations, and I never lectured on it. So I want to talk about that really quick also, because that's a very important subject that's going to come into play when we talk about evolution and stuff. So we're going to start with that first. We're going to talk about DNA mutations. And DNA mutations are changes to the nucleotide sequence that are the result of errors in replication. As a result of errors during replication. Okay, so not only are the complementary base pairs sometimes a little bit wonky, and the wrong purine is pairing with the wrong pyrimidine or whatever, but there are safeguards in place. DNA polymerase rather is supposed to be able to go back and check to make sure that those are correctly paired up. But occasionally errors are made, and hopefully those errors would be caught during G2 checkpoint, because that's what G2 checkpoint's all about, is making sure that the DNA was replicated correctly. But occasionally it's not caught, and that gene line or that lineage of cells will all have a slightly mutated gene. Sometimes it matters, sometimes it doesn't. Okay. All of the differences that we see among us today are the result of something that are called single nucleotide polymorphisms, which are changes in just one nucleotide. Okay. Single nucleotide polymorphisms. Or SNPs are changes to only one nucleotide in a sequence. Sometimes single nucleotide polymorphisms can cause extreme changes in a gene, okay, and that's what we're going to talk about first. If you have a single nucleotide polymorphism that causes either a deletion or an insertion, that can cause a serious change to the rest of that sequence. Okay, then let's look at why. Deletions and insertions. Again, just a one nucleotide change here, but one nucleotide is either added or one nucleotide is removed. So an extra one got in there somehow, or one of them was missing. Okay, so let's talk about, let's do an example here where we put in a normal sequence. And in our normal sequence, let's say it's GCA, GATACA, GGG, or something like that. Okay, so our deletion sequence, let's say for the purpose of this, we're going to delete this A here from the first codon. So we're going to get GC, GATACA, GGG. Okay, and then for our insertion, let's say that we are going to have two C's at the beginning. So instead of GCA, we have GCC, A, GATACA, GGG. Okay, so now you can't really see anything of consequence yet, but when you go back and you actually map out which codons you've got GCA, GAT, ACA, and GGG. Okay, in this one, you've got GCG, ATA, CAG, and then GG question mark, whatever the next nucleotide in that sequence will be. Okay, and then this one you've got GCC, AGA, TAC, AGG, and then G question mark, question mark. So the point that I am trying to get across here, the thing that I want you to notice is every single codon has changed. Even though we only added or subtracted one nucleotide, it changed every single codon after that, right? If every codon after that is different, that means that every amino acid after this is different. Which means if you change every single amino acid in a protein, is that protein going to resemble the previous protein at all? It's going to be totally different, right? 100% different. So this is the result of something that's called a frame shift. Okay, so frameshift mutations are mutations where every single codon after the mutation is affected. Yeah?
Why does every codon have to change?
Because if we're doing groups of 3, and we change the first group of 3 into a group of 4 or a group of 2, then every other group of 3 after that is either going to be one before or one after, like one before where it used to be or one after where it used to be, right? So every codon after the mutation changes. Okay. It's for this reason that deletions and insertions are generally non-functional. Okay, you will probably not get a functioning protein by having a deletion or insertion. Okay, what if you do? What if we just randomly put together 200 amino acids, because they're all different now, so we're just talking randomness. And what if that makes a functional protein? First of all, it would be amazing for that to happen. Like we're talking, this is 0.00000000001% of the time that this could actually happen. But this is going to create a new, novel protein, one that's probably going to do something that has never been done before. Okay, it's going to catalyze a certain reaction. It's going to be a pigment that absorbs colors that have never been absorbed before. It's going to do something new and different. The odds of this happening, very small. Let's say that one of your liver cells has one of these mutations that makes it awesome. It happens like right now, like boom. You've just had a liver cell mutation that makes it do something awesome, like produce a new enzyme that's going to help you out greatly in digestion or in the breakdown of blood cells or something like that. Okay? Let's say that just happened. Let's say it helps you during your lifetime. Then what?
You pass it on to
We normally would think we'd pass it on to our offspring. But are your liver cells going to be passed on to your offspring? No. Spermatocytes, oocytes. Those are the only cells that you could have a mutation in right now that would be passed on to your offspring, right? That means that not only does this 0.00001% chance have to happen, but it also has to happen very early on in your development. It has to happen in like the zygote or in the 8-cell stage or in the 16-cell stage to the point where it gets propagated throughout your entire body and then would be present in your spermatocytes or in oocytes, right? Which makes it even less likely to happen. But we see this happening fairly often in prokaryotic cells. Why is it more likely to happen in prokaryotic cells? Yeah.
They divide more rapidly.
They divide more rapidly.
They don't have like sex cells.
They don't have sex cells. They're asexual. So that means 100% of their mutations are going to make it into the next generation. So we can easily observe mutations going on in prokaryotic cells, whereas very, very unlikely that we'd see a new novel mutation occur in like the human race and then be propagated throughout the human race, right? That's very unlikely. All right. Let's talk about the types that we do see, though. The types that we do see are substitutions. Okay, substitutions are when you have just a single nucleotide change. A single nucleotide is substituted for another nucleotide. Okay, 3 possible outcomes can happen here. Outcome number 1 is probably the most interesting to consider. It's called a silent mutation. Okay, a silent mutation is where the mutated sequence of DNA makes the exact same protein as the non-mutated sequence. Okay, let's look at why this can happen. This is a DNA codon chart, so you'll notice there's no U's on there, there's T's instead, right? But let's say that we had the sequence CTT here, okay? Let's say that the last nucleotide in that sequence got substituted, and now it was CTC. It still codes for leucine, right? And that means that even though the nucleotide sequence has technically changed now, it's still coding for the same amino acid, and therefore there's no change in the protein and no difference for that organism. Never ever would you notice that because it's the same exact protein. Okay? One of the very interesting things that we can do with this is we can look and see how related organisms are. Okay? So if we take a protein that is vital for life, one that most organisms have. So if we look at like eukaryotic cells, we can say, okay, all eukaryotes have mitochondria, and mitochondria have sets of proteins that are required. One of the ones that we always use is called cytochrome C. It's a protein that's embedded in the mitochondrial matrix and is used in the electron transport chain. So we would think most living things, most eukaryotic cells are going to have the exact same cytochrome C because it has to be shaped a certain way in order to function. Okay? So we can go through and we say, okay, let's sequence the nucleotide sequence for chicken cytochrome C, and then let's compare it to human cytochrome C. Okay, and we might see, hey, look, the proteins are exactly the same, but in the time where chickens and humans have diverged, they have accumulated 20 silent mutations that make their nucleotide sequences different. Okay, so we can say, okay, 20 differences between humans and chickens. We can sequence chimpanzees, the exact same sequence, cytochrome C, and say, okay, humans and chimpanzees only have 3 differences, but humans and or chimpanzees and chickens have the same 20 differences. And so this tells us that humans and chickens diverged to a point that was longer ago than humans and chimpanzees, and that the common ancestor that diverged there was the same common ancestor that then diverged to form humans and chimpanzees, right? Because we both have 20 differences from chickens, but we have 3 differences from each other. We can also determine that the divergence was closer in the geologic time scale for humans and chimpanzees than humans and chickens. We can take chickens and chameleons, say, right, and say, okay, let's look at that. Let's say chickens and chameleons only have 8 differences. So we know that chameleons and chickens diverged relatively more recently than humans and chickens diverged, right? And so we can use this to sort of map out this tree of life and say, this is the relative time length that that everything is kind of branching off. Okay, so anytime you see one of those tree of life things, the way that we estimate the length of time in which they branched off is A, through fossil records, and B, through the number of mutations in a common gene like cytochrome C, number of silent mutations. All right, the other type is called a missense mutation. And a missense mutation is going to change just a single amino acid. So one amino acid changes. All right, so if a single amino acid changes, that means that the protein shape overall will change, but not so drastically like we saw in a frameshift mutation, because most of the amino acids are going to be exactly the same. We're just shifting one of them. So the protein might still be functional, but just slightly less functional, right? Might be slightly more functional. Who knows? All of the differences that we see among us right now, the differences between your height, skin color, hair color, eye color, all those things are all caused by missense mutations, substitutions where one amino acid changes and the overall protein shape changes slightly. Okay, still functions most of the time. In the case of blonde hair, not so much, but still functions most of the time, but just functions slightly differently. Blue eyes, green eyes, brown eyes. That's what we see there. Okay. All right. The last one is called a nonsense mutation. And a nonsense mutation is when a nucleotide is substituted in a start codon or a stop codon. So if you remember how translation works, a ribosome attaches to the G cap of an mRNA strand, and then it goes down, it translocates down to the first AUG. Okay, it's looking for that AUG methionine start codon. Okay, if there is a substitution in that AUG, in that first AUG, then it's not going to stop at that point anymore. Okay, and it's not like methionine is only used for the beginning of a protein. It could be used in the middle of a protein also. It's a normal amino acid, right? And so instead of that ribosome stopping at that first AUG, it's going to continue to translocate down until it reaches the next AUG. Maybe it occurs 50 codons later. Okay, so if you were supposed to have a 200 amino acid long codon or amino acid long protein, now you have a 150 amino acid long protein, and so that's probably going to be a lot different in its function, right? Maybe it's going to be non-functional. Maybe it's just going to have a slightly different function now that it lost one functional part of it. Okay, so this is generally going to be much more detrimental than a missense mutation. Let's talk about it in a stop codon. Let's say you were supposed to have a stop codon after 200 amino acids, but now you don't, and so it's just going to randomly look for the next stop codon then, which could be in the middle of an intron, or it could be in the middle of the next, or at the end of the next gene. You might combine two proteins together here. You might have started out with a 200 amino acid long protein, but now it's producing a 2000 amino acid long protein, right? Giant proteins. That's probably going to be bad. It's probably going to be detrimental, and the chances of it being actually useful are so low. Okay? Questions on mutations?
Would the nonsense mutation only occur in one cell?
Yes.
Okay.
Yep.
So it wouldn't be like all of the variants.
Correct. And that's basically the case with all these. And most of the time when there is a large mutation that causes a protein to become non-functional, that cell is aware of it. We talked about all the like tumor suppressor genes and regulatory genes that go into this. And so that will trigger apoptosis if they're not producing an enzyme correctly that is required for the life of that cell. All right, so let's talk about the next thing, which is prokaryotic gene regulation. The first thing that you need to understand about this is that prokaryotes are dumb. They do not think. They don't have nervous systems. They don't have brains. They don't make decisions. They don't have a conscious decision-making process, right? So for me, I have a skill set. We all have fairly large skill sets, even if you think you don't. You do. I can do many things like swim and run and breathe and play the guitar and things like that. And I decide, using my brain, when it is appropriate for me to do various things, okay? For instance, right now, it is not appropriate for me to be swimming because I'm not in water, and if I was just swimming around in the hallway or the classroom, they might remove me from those areas thinking that I'm a crazy person, right? Similarly, if I'm sinking in the ocean, it would be inappropriate for me to play guitar at that point because I would then die. Okay, so I can decide when it's appropriate for me to use things in my skill set. Prokaryotes have a skill set, but they cannot decide using their brains, because they don't have brains, when it is appropriate for them to do various things. And so they rely on these things that are called operons. And every single thing in their skill set has an operon, and they work like this. At the very front, there's a promoter. Then there is a area that's called a regulatory gene. Then there is another promoter. Then there is an area that is called the operator. Okay, and after the operator, there is a functional or structural gene. Okay, so the way that this works is that we have normal protein synthesis going on here. This is going to represent RNA polymerase. Okay, and this RNA polymerase is going to move around and attach to the promoter, right? And as it attaches to the promoter, then it's going to transcribe this regulatory gene. That's going to be made into mRNA. The mRNA then gets made into a protein. It's going to attach to this promoter here, and it's going to go down here and make this into mRNA. That's going to be made into a protein, right? We're all familiar with how this works? Yes. Okay, good. The difference is that once this goes and attaches to the promoter, it's going to produce this regulatory gene, and the regulatory gene produces a regulatory protein, and the regulatory protein is one that attaches to the operator. This is called a regulatory protein, and sometimes called the repressor. Okay, so you can probably see, hopefully, what happens now. If you've got RNA polymerase, it's able to attach to this promoter freely. It's able to create this protein from the regulatory gene, right? But over here, it can't get to this promoter because there's something in the way. There's an operator, and the operator has this regulatory protein on it, the repressor, that prevents the RNA polymerase from attaching to it. Okay, so let's take an example of this. Let's say that this gene here produces lactase. Lactase is the enzyme that breaks down lactose. All right? Do you think that it is useful for a bacteria to have in its skill set the ability to break down lactose? Yes. It's a sugar. It's a disaccharide. So in itself, that disaccharide can't be used. So it has to be broken down into monosaccharides before it can be used by the bacteria. So it would be good if, in the case where this bacteria is exposed to lactose, that it could then break it down. All right. However, let's say this bacteria is on my desk. There's lots of bacteria on my desk right now, all over the place. It is probably not going to be that important for this bacteria on my desk to be producing the enzyme lactase right now. Very much like it's not important for me to be swimming right now. There's no reason, there's no lactose on my desk. I haven't poured milk on my desk recently, right? And so there's no reason for it to be producing that, all right? In fact, it would be a big waste of time and effort and energy and amino acids, and it would be just a bad idea. But in the case where there is lactose present, so let's say now there's lactose present, that's going to be this stuff. This lactose can go and attach to the allosteric regulation site, remember that from Unit 1, of this regulatory protein, and that will, in turn, change the shape of the protein. So maybe now it's got like little Bart Simpson hair. And it does this whenever it's got lactose attached to it. Okay? This is the inactive form of the protein, meaning this protein can no longer attach to the operator here. Okay? So I'm going to erase mine, but you don't have to worry about it, right? So I'm going to erase it just for visual purposes. So then now this RNA polymerase can go around, it can attach to the promoter, and it can make this functional gene. It can produce the mRNA for it. That's going to produce the protein lactase. The protein lactase is an enzyme that's going to break down lactose. So what does that do? It breaks down this lactose, it breaks down this lactose, it breaks down this lactose. What happens when this breaks down this lactose? It then becomes active and begins to repress this gene again because there's no more lactose present. And the next time lactose is present, then it will start producing this lactase gene again. Right? See how this works? It's able to respond to its environment without having to think about responding to environment, just by chemical reactions. Okay? This is called an inducible operon. An inducible operon is one that has a default state of being repressed or being off. Okay? So it's an inactive default state, which means that the regulatory protein is attached to the operator. Is normally attached to the operator. Okay? And because it's inducible, it is able to be turned on in the presence of a certain chemical. In this case, lactose. So our example here is the Lactase Operon. Okay, this is in contrast to the other type of operon, which is called a repressible operon. You can probably figure out what that is based on the previous definition. If an inducible operon is one that's normally off, repressible operon is one that's normally on. So this has an active default state. Meaning that the regulatory protein is normally inactive and not attached. Okay? So if it's repressible, that means it's able to be turned off. By the presence of a specific chemical. Okay, and a good example of this is something that's called the Trp Operon. And the way that works, Trp is the abbreviation for the amino acid tryptophan. And tryptophan is a relatively uncommon amino acid when we look at the 20 amino acids. You can sort of look at the chart of amino acids here and say like, oh, leucine's probably pretty common. But if you look over here, tryptophan is only coded for by a single codon, TGG, right? And so it's fairly uncommon. But that's not to say it's not important. You still have to have it to produce a protein that has tryptophan in it. And so it actually is one of those amino acids, like many of them on here, that can be produced by changing one amino acid into another amino acid. There's an enzyme, a pathway, a chemical pathway that will change a single amino acid, one type of amino acid, into another type of amino acid. So, what's up?
Is that whole thing like Operon?
Yeah. Oh, yeah. So I probably should have said that. This whole thing here that includes both the regulatory gene and the functional gene, that's called the operon. Thank you. Okay, so let's talk about tryptophan. If the cell is in a medium that doesn't have any tryptophan in it, it needs to produce its own tryptophan. So let's say that this functional gene over here produces an enzyme that's able to catalyze a reaction that converts one amino acid into tryptophan. Okay, so normally it's on. And it's on because it's in an area there's no tryptophan, okay? But let's say that the cell has produced a lot of tryptophan and it's not using it. There's 500 tryptophan amino acids floating around that are not being used, okay? Those tryptophan molecules can then attach to the regulatory protein, but since this is the opposite, instead of turning it to its inactive state, it's going to turn it onto its active state, which then makes it go and attach to the operator, which then blocks the production of that enzyme that makes more tryptophan, which then stops more tryptophan from being produced because there's plenty of it, right? So it's working in the opposite direction. This is this is inducible, but it would be if we changed it so that the active state was the one that had the molecule attached to it, that would be in a repressible operon. Does this make sense? Understand how bacteria are able to respond to their environment and decide which parts of their skill set they should be using at any given time without having brains? Yes? All right. That's all I got.